What if we could write a better story for patients with kidney disease? We're excited to see where that question takes us. Curiosity has really driven Arcana from the beginning. What if we could provide same-day service? What if we could improve patient care through clinical trials? What if we could figure out the driver of these diseases? When Dr. Walker started Arcana 20 plus years ago, he was really trying to reduce the amount of time it takes to get the results from a kidney biopsy. It used to take a couple weeks. And if you've ever gotten anything done in a hospital, you want those results immediately. And so that was the main goal, was to reduce the time to get somebody an accurate pathology result from their kidney biopsy. Nobody thought it was possible to do a same-day biopsy service, but he made that possible. He pushed the envelope, and he was able to provide that for patients, which was a huge advancement in the field and has pushed everyone around the country and around the world to reduce their wait times. So what used to be weeks can now happen in a single day. That's important for many reasons. It reduces patient anxiety because they get that result quickly. It also can have a huge impact on patient care. For certain diagnoses like ANCA, anti-GBM, we're getting the result to them much faster than they even get serologic testing done. And so they can start treating a patient immediately. So they are so happy and grateful for that, because the difference in a day or two of turnaround time could mean the difference between losing your kidney and saving it, keeping that patient off dialysis, off the transplant list. So it can make a huge difference in patient care. It's unique that we have so many people dedicated to renal pathology in one place. There's very few institutions that have more than one renal pathologist. So the fact that we have 20 with such a wide breadth of experience is really unique to Arcana. We are continually sharing our observations, sharing interesting cases, and out of these daily discussions have come real advances in renal pathology. We've noticed certain patterns of disease or certain ethnicities in the link to disease, and that has led to huge advancements in discovery of new genetic diseases, of naming patient diseases so that patients really have a name to put to what their kidney disease is. And when you can do that, that opens the door for research into specific diseases, understanding of treatments work in different disease states, and then that gets people excited to research those questions and look into similar cases and see what the similarities are. And those daily discussions have spurred very interesting research questions. We all have this curiosity inside of us that leads us to ask those questions and want those answered. So at Arcana, we're not required to do research, but we're really driven to do research. With over 20 years and over just hundreds and thousands of biopsy samples, we feel that it's our responsibility to do this research that might not be possible elsewhere. We have access to rare diseases that are only seen a few times a year and make advancements in those areas. And we feel very passionate about expanding the access to renal pathology education. As a group of over 20 renal pathologists, Arcana Lab has a unique ability to give high-quality educational content out to the world, something that you might not have access to at your individual institution. So you can listen to excellent podcasts, educational materials, lectures, tutorials. We want everyone to have access to this information to help improve patient outcome and patient care. We want to empower patients with knowledge about their own kidney disease. Arcana has changed a lot in the last 20 years, from providing the fastest possible kidney biopsy result to becoming a hub for clinical trials, contract research, and education, all in the aim of providing the best possible patient care.
What if every biopsy which we perform can be assessed at the bedside at the time of the procedure, and a smartphone app can tell us that we did a good job? So every dream which we have in the field of nephrology, that future is dependent on the biopsy. So if it starts with kidney biopsy, then kidney biopsy needs to be adequate. When a patient walks to a clinic to get a treatment for a kidney disease, for some of the diseases we need to do a kidney biopsy to render the correct diagnosis and the correct treatment. We take a biopsy, we send that biopsy to the lab, we get a diagnosis, and we treat the patient. we assume everything is going to go smoothly. But there are 2% to 14% of the patients who are not going to get that because the biopsy was inadequate. If we do our job right, if we can assess the adequacy of the biopsy just at the time of biopsy, those 2% to 14% of the patients are also going to get the right treatment at the right time. When you listen to the word biopsy, it sounds like a simple procedure, but for a kidney biopsy, a patient needs to go to the OR. It is a big procedure. And for some kidney diseases, sense of urgency is very important, like you need to start the treatment now. So in those situations when we call the clinician and let them know that, hey, you got all medulla and I won't be able to render the diagnosis, the stress and the pain the clinician goes through is completely unimaginable because now they need to call back the patient, they need to reschedule the biopsy, the treatment has got very prolonged, they won't be able to start the treatment on time. And now a patient has to go through that pain of biopsy again. So our dream is to have a smartphone which can assess the adequacy at the time of performing the biopsy, so that all the patients are going to get the right diagnosis at the right time so that they can get the treatment at the right time. We need cortex when you biopsy. When you go inside, take a biopsy piece, did you get the cortex? If you have got the cortex, that is an adequate pass. We started a workshop just to teach kidney biopsies along with our nephrologist friends, and we do that at Kidney Biopsy Academy at KidneyCon. We have also published papers. We have also spoken to a lot of nephrologists and radiologists so that they can improve their procedures. So now there are many places which have seen significant difference in the rate of adequacy, improved from more than 25% to less than 2%. And we are also pushing that understanding that the adequacy can be assessed at the bedside using a smartphone. We talk about research. We talk about advancing the field a lot. That has to start somewhere.
What if transplant rejection could be routinely classified based on the molecular pathogenic drivers of disease? That's exactly what we're trying to do here. It's really important for us to be able to extend the life of every one of these transplant organs. It's important for the patient, it's important for the community. These are precious resources. So we need better diagnostic assays in order to know how to treat these patients more effectively going forward. In 2019, the BAMP Foundation endorsed a gene panel for the transplant field. It's known as the BAMP Human Organ Transplant Panel. It's usually abbreviated BHOT. And this BHOT panel consists of 770 genes that were selected based on their relevance to transplant organs and transplant rejection and diseases of transplants. So in addition to putting that panel together, they also selected a platform to run it on. And they selected the NanoString platform. And it was selected for two primary reasons. Number one, the NanoString platform is widely available throughout the world. But number two, the NanoString platform is particularly good at quantitating gene transcripts from formalin-fixed paraffin-embedded tissue. So that's the same tissue we use for light microscopy. And the reason that's important is that means that we can take that tissue and we can analyze it for molecular classification of the biopsy as well. We took this setup, this BHOT panel running on a NanoString platform, and we used it to analyze more than 1,400 kidney biopsies. Most of those were transplant biopsies. And we took the data from that analysis and we used it to build a multi-class model for the diagnosis of transplant rejection. So now we can take a transplant biopsy and we can put it through this classifier, and it will classify with high accuracy the type of rejection or lack of rejection within that biopsy based on this molecular classification. In the near term, I think molecular classification is going to serve to kind of risk stratify these patients and kind of add another layer of information to what we're currently doing. But long term, the ultimate goal is to be able to identify the root cause of a patient's rejection at the molecular level so that we can offer therapeutics specific to that patient's rejection. So we're trying to move towards the day when we will be able to truly offer precision therapeutics for patients with transplant rejection. This whole project went from an idea to where we are today in just over a year, which is pretty remarkable for a project of this scale, and it just would not have been possible without a dedicated team from both Arcana and CareDx.
What if you woke up and there was no need for clinical trials? What if we had cures for renal pathology? We work with our partners and our sponsors every day to try to put ourselves out of business. Here at Arcana, we have the opportunity to partner with biopharma companies from all over the world. We have the capacity to work with them to assess not only the mechanisms of renal pathobiology from common diseases all the way to the most rare diseases. Arcana loves to partner with companies that are particularly looking at these rare diseases, so-called orphan diseases that can affect, in some cases, less than 1% of the population. And given the large data sets that exist at Arcana, we really feel that we have a responsibility to work with these sponsors and to assess not only new therapeutics, but also the mechanisms that underlie, and really find out why do people suffer from these diseases and what can we offer to treat them. The volume of renal biopsies that we see at Arcana are really unmatched. So even uncommon diseases are well represented within our data sets. I've spent my career really working in drug discovery, and very, very few compounds ever go on to actually become a drug. But here at Arcana, we get the opportunity to work with biopharma sponsors, looking at all kinds of diseases. So we really have a wide breadth to impact patient care.
What if we can use the molecular pathogenic drivers of the underlying disease to improve our diagnosis and prognosis of lupus nephritis? I really think that we can identify more from a kidney biopsy than what the disease is at that snapshot of time, and that proteomics is going to help us find biomarkers, but hopefully not only to monitor disease, but to inform treatment. So I'm really passionate about studying lupus nephritis because I really cared about someone who had lupus. I had a close friend and colleague during graduate school who had the disease. And during her 20s, I saw the effect that it had on her. In her early 20s, she required bilateral hip replacements from chronic steroid therapy. She was chronically tired, particularly when requiring dialysis. It really takes a lot out of you. And unfortunately, had succumbed to the disease. Unfortunately, her outcome isn't unusual among lupus patients. For patients requiring dialysis, there's approximately half of them will die in 5 years without a transplant. From patients with lupus, about 50% of them will develop nephritis at some point in their disease course, and about 1/3 of those patients will go on to develop end-stage kidney disease. It's a disease that can flare and remit, and therefore over time there can be chronic renal damage occurring. So one of our main hurdles in improving our understanding of lupus nephritis is that we incompletely understand the disease pathogenesis. Another significant hurdle is that it's a relatively rare disease affecting only about 2% of patients with kidney disease. However, here at Arcana, we have the unique opportunity to partner with multiple nephrologists to study larger cohorts of patients to make that possible. I believe that proteomics, in conjunction with histopathology, will be the future of lupus diagnosis, and hopefully can provide some information of some proteomic drivers of disease and potentially biomarkers to follow. So currently there are multiple clinical trials in lupus nephritis looking at a multitude of biological targets. One avenue through which kidney pathologists can help nephrologists is formation of companion diagnostic assays to be able to identify who may have activation of those particular pathways of the disease. So one of the big questions that we have with lupus nephritis is, can we get more information out of the diagnostic kidney biopsy than just what the disease activity is for the patient a snapshot of time? To investigate this, we're using proteomics, or using mass spectrometry to look at unique proteins on lupus kidney biopsies. And we hope that this could inform both their prognosis, identify those that are at high risk of progression of lupus, as well as to identify what are the key pathogenic drivers in individual patients that may be driving their disease and ultimately potentially lead to a choice of therapy. For some types of lupus nephritis, a certain antigen type is identified, of which then could lead to formation of non-invasive tests to hopefully be able to monitor that disease.
What if the Multiplex Imaging Center at Arcana were the leader in innovation and invention in renal pathology and understanding of inflammatory disease in the human kidney? I mean, as a renal pathologist, one of the reasons I show up to work every day is to be able to answer the questions one day that we can't answer right now, such as how should I treat this patient, what's going to happen next. Multiplex imaging is a term that is fairly broad, but really it comes down to multiplex, which is sampling multiple things at the same time, and imaging. So we sample multiple biomarkers or multiple types of molecules within the tissue by imaging, and then from that create a map of where those molecules are, whether they be proteins or small molecules or even transcripts. To create neighborhoods of the tissue, a map of where those are, to then correlate with outcomes. So as a way to really just make further measurements and further refined measurements of the tissue. We can make better use of the resource we have. I always think of it as making treasure from the trash. So can we take surplus diagnostic specimens, things which have run their purpose for diagnosis, but still retain all this rich information about the disease process? Can we use that to better understand what's happening in the patient? We're just scraping the surface with the types of information that we derive from tissues for diagnosis. And certainly we can derive all of the information that we need for a diagnosis at this point, but there is much more that the tissue has to tell. There are many more processes that are happening which we fail to detect with traditional diagnostic methods. So what we hope to uncover are not only the deep phenotypes of the cells which are involved in the processes there, but also what are they doing? How did they get there? What are they going to do next? I would envision multiplex imaging to be applied to improving diagnosis and therefore patient care by having some predictive biomarker panels for inflammatory diseases in the kidneys, particularly my interest in transplant rejection, but also lupus nephritis or IgA nephropathy, sort of medical renal diseases affecting native kidneys. One of the first research questions that we're deeply investigating using the resources of the Multiplex Imaging Center at Arcana is to better understand T-cell mediated rejection. Specifically, can we derive predictive biomarkers of treatment resistance from the tissue taken at the time of diagnosis? In order to move multiplex imaging from bench to bedside, I think we really ought to develop closer partnerships with our clinical colleagues, as well as colleagues who have access to other deep dive technologies to interrogate the tissue. So using the tissue and using multiplex imaging, we can take a really deep dive into understanding what the cellular players are within the tissue. But then it really does require a correlation to real-world outcomes. And I think that for that we'll need strong partnerships with our clinical collaborators.